Hey, it's Valerie Estes and Erin Fleming coming to you guys live with this week's Project ALS prosetin update. Prosetin is a promising new ALS drug candidate that we aim to deliver to first patient trial in 2020. As you know, we're excited about prosetin because it will be available as an oral suspension. It bathes the brain, and it's a therapy that's designed to reduce cell stress implicated in ALS. Once again, prosetin is orally available. 2, it crosses the brain-blood barrier. 3, it is designed to engage a specific therapeutic target in ALS. I'm going to be honest with you. We've lost some time on prosetin 2020 because of the COVID pandemic, but Project ALS is working hard to minimize our losses. We're minimizing losses in large part because of today's guest. A member of the Project ALS prosetin dream team, Dr. Lauren Black, specializes in drawing a straight line between 2 points, between dire human need in ALS and other life-threatening diseases and hopeful new therapies. She gets it done, and you'll meet her in a moment. First, happy Cinco de Mayo, everyone. Happy May. This is ALS Awareness Month. Today is also Giving Tuesday, and you can help us move prosetin closer to patient trial right now by going to prosetin2020.org. That's prosetin.org. Give what you can. And this is really the cool part. Today, the Project ALS Board of Directors will match your generosity up to 10K. That's a lot of money, right? But today only, okay? So the magic happens. You give $10 and it turns into $20. It's just like magic here. You give 50, abracadabra. We're looking at a C note for prosetin, okay? So come on, guys. The Project ALS board urges you to get involved. We really need your generosity to kick in, and they'll match it right now. So for today only. So please head to prosetin2020.org. Today is personal for me. My sisters and our friends and I started Project ALS over 20 years ago when my sister Jennifer was diagnosed with ALS at the age of 35. We've put a lot of research progress on the map, but perhaps our biggest achievement at Project ALS has been venturing outside ALS borders, recruiting deep talent, intelligence, experience, and innovation, and sort of hauling it back to ALS research. Today's guest is a shining example of the Project ALS mission and its success. We read about her about a year and a half ago. It was online, actually. It was a story about out of the laboratory of Dr. Timothy Yu at Children's Hospital, Boston. Dr. Yu and Lauren Black had worked to provide a first-in-world treatment to one of Dr. Yu's patients, a little girl who was very, very sick. Dr. Yu had developed a long shot of a therapy for this girl, and Dr. Black helped to move that long shot to a first-in-world trial for that girl. So Project ALS decided to call Dr. Black straight away. We asked her if she couldn't help us do that in ALS, to draw that straight line between 2 points, dire human need and the dream of treatments. It's just an honor to welcome you to the live stream, Lauren and Erin. Why don't we get it going in style? I'm kicking it to my buddy, Erin Fleming.
Thanks, Valerie, and hi, everyone. I think as Valerie said, all of these webinars are really special to us at Project ALS, the opportunity to get experts who are helping us to bring prosetin forward and to communicate, hopefully, in a helpful way to everyone about what this process actually looks like. But as Valerie said, today is really special because we just have, we reached out to Lauren, and she's really changed so much for how we as Project ALS have thought about being able to approach drug development and just the world of possibilities for, as Valerie said, drawing a straight line between 2 points. So before we kick it over to her, I just want to give her a proper bio, which is, so impressive. How could I not? Lauren E. Black, PhD, is a distinguished scientist at Charles River. She has more than 25 years of experience in drug development, focusing on accelerated translation to phase 1 and 2 clinical trials. She mainly works at the first-in-human stage at high-risk diseases using novel products. And she's been a vital partner to us in this role on the prosetin team and in many other capacities. For prosetin, she has advised design and oversight of our non-clinical toxicology program and our entire regulatory strategy. Dr. Black holds a BS from Carnegie Mellon University in molecular biology and a doctorate in pharmacology from the Virginia Commonwealth School of Medicine. She spent 2 years in postdoctoral research at NIH, evaluating dopaminergic drugs and GPCRs. What does that mean? She'll explain more. Prior to Charles River, Dr. Black served as a reviewing pharmacologist in the FDA Center for Drug Evaluation and Research and the Center for Biologics Evaluation and Research for 11 years. She focused on the preclinical assessment of transplant drugs, oligonucleotides, monoclonals for autoimmune diseases, vaccines, and cell therapies there. And since 2002, she's worked at Charles River in the Scientific Advisory Services Group. And she sits on several government, academic, and industry advisory panels where she focuses on important issues around in vitro toxicology, portfolio prioritization, technology transfer, and regulatory strategy. So, just a wealth of knowledge that she brings to us. And that bio, it's very impressive to me, but I'm not really sure what it means. So maybe, Lauren, you could start off by just explaining how you ended up becoming a distinguished scientist at Charles River and sort of this path that you took to get there.
Sure thing. I just wanted to say, first off, I'm really honored to be here. And you guys have been some of the best people to work with I've ever worked with. Really focused on the patients and bringing new therapies forward, working with the academics at Columbia and other places, University of Massachusetts and all the other folks you brought together. And so, I think you should be applauding yourselves for a lot of this progress and straight lining, so back to you. But I think, it always takes a village to bring a drug forward. And the first thing you need to do when you're on a road that you don't know where it goes is to ask directions. And that's where you guys have been key, is pulling out that roadmap and saying, are we going the right way? What else do we need to bring in here in terms of experience? And I think that's really all about what drug development is today. It's not about somebody who invented something like a light bulb and just turning it on and then saying, okay, everybody needs to electrify. Right. There's a lot of infrastructure behind drug development. It takes a lot of people to really make it happen. And they're not going to be all at one place and at one time. When Charles River does drug development, we bring in, I was just on a TCon with 18 people from Edinburgh over to Reno, Nevada. And we had chemists and biochemists and toxicologists, multiple of each type of person on the call to try to get a quorum on exactly what to do. It's just not about one idea anymore. It's about multiple people and all their ideas synthesized. And that's what's going to make it happen. It's like a democracy out here.
Yeah.
In the drug development world.
So Laura,
it makes the patients have to be part of that, what they want to see happen for themselves.
Yeah. So Lauren, when you're on that call and it's a village, what are you doing in the village?
Sometimes it's herding cats, other times it's about providing guidance at a senior level. You mentioned I'd worked at FDA a number of years. I also worked as a basic researcher when I was in NINDS doing neuronal research. So I was a neuroscientist at one time, pipetting into the laboratory with, scientists don't really have Erlenmeyer flasks with red and green liquid behind their heads like they have them in the movies, or they don't have weird hairdos. We really just work long hours pipetting clear liquids into other clear liquids and hoping that we did the right thing with the right markings on the vials. And then at the end of the day, you get your research back and you try again. So I've done that work. I did it 18 hours a day for a year and a half. And then I went over to FDA, and it was in the middle of the AIDS epidemic, which is not too different from the kind of panic that we have right now with COVID, and the kind of panic we should have about diseases like ALS. I mean, we don't have but a couple of drugs, and everybody knows we need far better interventions for ALS that not be disappearing from the radar. It needs to be right up there with cancer. It needs to be up there with neurodegeneration in children. It needs to be right up there with all the other priorities we have. There was a big fluff about, what was a decade ago, the decade of the brain. What happened to that? It kind of disappeared in all of the other priorities that we had. And we need to get back to taking the power for these very, very aggressive diseases that are taking over our lives of so many people. What do? I help at a senior level in our company. I help provide some of that academic experience, and also, the 25 extra years I've got doing drug development on high-risk diseases. There's a lot of research that you actually don't need to do to move drugs forward. So sometimes it's as much about what you decide to do the research on and what you decide not to do. Like if you're doing a research paper and you have 4 postdocs in 20 years to write a book on a new basic mechanism in the nervous system, you have a lot of things you need to cover in depth and in breadth. But when you're developing a drug, it's much more applied research. You need to focus on the goal, that straight line thing. And you need to say, okay, how much work do we not have to do and still move this into humans? So there's a certain amount of, it's kind of like going into a garden and pulling the weeds. You've got to really just let the plant grow, get enough weeds away from it. But if you spend all day, you're always going to have another weed to pull, right? So you just got to go in there and say, I've got an hour, I'm going to weed away from the plant so it can prosper. That's sort of the mentality you put towards drug development. You say, what exactly do I absolutely have to know this month and in 6 months? And then sort of back plan all your experiments accordingly. It's very practical.
Yeah. So maybe we can get into the weeds a little bit. That, wasn't the natural segue. But you are helping us to sort of create this program for prosetin, where we're trying to move as quickly as possible and to only weed what we need to weed. I think there's a big question that I certainly had when we came into this, which is, in a disease as serious as ALS, people have expressed, and I understand this very deeply, there's a much higher risk tolerance. It's like, what's the outcome of the disease without a drug? Why can't we just try drugs immediately? And so I wonder if you could speak to, this sort of balance. Why do you even need to study the safety of a drug that's going into a universally fatal disease? What do you actually get out of these IND enabling studies? And how do you think about that?
Well, I mean, a lot of people have either, they don't understand necessarily how toxic drugs can be. I mean, honestly, if you look at the label for Tylenol, you probably wouldn't take it. If you take, I don't know how many doses it is, don't quote me on this, but if you take a look at the Tylenol doses that can kill your liver, it's not very many pills. One of the leading causes of liver transplantation in children is a child that breaks into their parents' Tylenol bottle. It's one of the most toxic drugs in your typical medicine cabinet. As we all know, you don't give aspirin to children either.
Right.
So here are 2 drugs that have been around for God knows how many years, and yet they're over the counter, and yet they are extremely toxic, particularly to certain sensitive populations. Kids being one of them, geriatric people to the other side of the equation, women of childbearing potential. It's a long word for, younger women who are still having kids. And I think that we have to be cognizant that not all of the actions of a drug are visible.
Right.
So when we design toxicology studies, we actually do a deep dive, and I know a lot of people have approach avoidance to this, but we have to do pathology on the animals, because we don't learn those kinds of events at the microscopic level in our tissues from a human. Neither you or I want to be donating samples of ourselves. So we need to see the unseeable with a drug. Some of the things are visible in terms of blood chemistry changes and behavior, and some of the changes are invisible to the eye of the observer. So when we do a tox study, it's with that, both of those things in mind. And the fact of the matter is that everything is toxic at one dose or another. We have to learn where the doses are toxic in terms of the exposures, so that we can go below those doses and safely dose people, and we don't make disease processes worse. There are many things about the body we don't understand yet, and those mysterious processes are often unseen. Like I said, clear liquid into clear liquid. You hope you understand what's going on in that biochemical solution. Once you put a drug into the body, you don't know exactly where it goes.
Yeah. So
where it builds up, you know?
Right. I think you've spoken to this a little bit, but what is sort of the critical information that, you know, regardless of the disease or the seriousness or how quickly you're trying to move, that you want to get out of these toxicology studies? Like, what do you want to know? What questions do you want to ask about a drug before you give it to a person, so that you can observe that when you do actually give it to a person?
Well, there's certain drug classes have known bad actor effects, right? Valerie talked about her sister. I can say, as an example, my mom. She died of a nasty brain bleed that resulted from taking warfarin or Coumadin, right? She had a heart valve, and so she had to take blood thinners, else she'd get a clot on that valve. Well, that's a classic case of the dose makes the poison. And in that case, you have the monitoring tool of being able to check the clotting potential of the blood, because you can actually take a blood sample and let it clot and count how long it takes on the countertop for it to form a clot in vitro or in the test tube. And you can say, well, if it takes X number of minutes to clot, you know that you have a certain amount of anticoagulant effect. So that's what we call it pharmacokinetics. We know the drug level, and pharmacodynamics when we know what the action of the drug is measured at, or the other word for it is a readout. So we can say exactly what the correlation of at least what's in the blood with what exactly the drug is causing. And in the case of Coumadin, we want there to be an anticoagulant effect, right? But what happens when you've got too much of that? A brain bleed,
Yeah.
right? So a little bit of a good thing is good, but then too much of it can be what we call exaggerated pharmacology, or it can be frankly toxic. So we all might like a glass of wine, but we all know that alcohol is toxic at too high a dose, right? You can kill yourself with that. So I think it's very important to understand this exposure response curve, whether it's a readout or the fancy words, pharmacokinetic, pharmacodynamic readout. And I think that that's what we're really getting at when we look for things that we can translate to humans from the animals. We can say that we understand these processes in the animals before we dose the humans, so that we don't, frankly, hurt people.
Right.
And lots of drugs are killed early in these stages and never makes the newspapers because it's private. You know, people are doing drug development under confidential arrangements. And so a lot of the toxicities are never discussed in public. They're just quietly shelved and a different chemical is tried.
Yeah,
So
no, it's a really good.
A lot of things you don't hear about.
So I guess, my lay takeaway is that you obviously need to do something. But for a disease like ALS, are there ways to move these programs forward faster? Are there sort of the weeds that you don't have to pull, that you can still move it forward? And are there things specific to ALS that you've encountered, that help you to accelerate these programs?
I think there are different subcategories, as we know, of ALS, and they vary from people that have really, really long and relatively active lives. And then we have all of the aggressive versions. And we've both worked with J.C. Hermstead lately, and she had a very aggressive form that's driven by genetic mutation in the FUS gene. And that's a very aggressive form that I came to know about from working with you all. It was a blessing and honor, by the way, to give JC our time at Charles River Laboratories. And we're sorry to hear of her passing. And I know that it is very difficult to cross-apply information from that case, but I think it does point out that the learnings that we undertake with very aggressive forms of disease are helpful in that they tell us we need to make the course of drug characterization fit the particular aggressiveness of the disease that we're working with. And if you want to say that ALS is many diseases, I think that's probably appropriate, right? We have many genetic conditions that can contribute to ALS. There are many different processes that contribute to ALS disease, including neuroinflammation, fundamental processes, and neurological processing. There are a lot of different kinds of drugs that could treat different kinds of ALS. So I think we have to break up the ALS categories into whether or not they're fast-moving and aggressive or more slow-moving, and then we can make the drug development process and the regulatory pathway fit that particular subcategory of patient.
I hear chants in the background. Hi, sweet doggy. You guys, I just wanted to
Let me
also
see if I can
mention
get him
that-
offline. Just a sec.
No, we want him online. We need
Okay.
chants.
Well, if he
But I
keeps
wanted to
doing it,
mention
let me
to
know.
Participants, if you have questions or comments for Dr. Black or either of us at the top here, please send them in. We want to hear from you. So
Yeah.
ask away.
I think, Lauren, your point that there are many different types of ALS is really well taken. I think the fact also obviously still stands that there are no treatments for any types of ALS that are really meaningful, that are FDA approved now, and it is currently still a universally fatal disease. So I guess, when we were talking about prosetin and trying to strategize for this program, which in theory prosetin is, it's a small molecule that could impact all ALS. This will be tested in clinical trials, but that's the idea behind it. Are there still sort of shortcuts, or maybe that's not the right word, but means to accelerate a program for a drug that's sort of for pan ALS, given the seriousness of the disease? Just sort of regulatory strategies or strategies in the preclinical program that allow us to move a little bit faster than for a disease that already has many treatments on the market.
Well, I'd like to bring up something that I wanted to say. The FDA does have the leading role in trying to define what accelerations are acceptable. We can always propose accelerations, but the FDA has to accept them.
Right.
And in order to do that, they have to be aware that, ALS is lethal and that it's very understudied and it needs to be pursued much more aggressively. They need to lower some of the burdens on the drug development process. And so I'm going to use my prop here. This is a recent guideline. I don't know if you can see that.
I can see it.
And it comes on, that's a typical FDA format. It has the date on it. It has this bold print they always use. And it says basically ALS, developing drugs for treatment, guidance for industry. And it's coming out it came out in September as a final guideline. And it came out from a committee that clearly met between two different, very different groups at FDA, the Center for Drugs and the Center for Biologics. So it represents all the people that would review gene therapies, oligotherapies, small molecule drugs, any kind of drug contributed to this guideline. It's heavily driven by the clinical team, I'm sure. So they did put their heads together, and they are saying that come and talk to us, come and propose those accelerations. They have a pre-IND process. And so for folks like me that work at the cusp between drug development, drug discovery and drug development, as you move into the first in human trials, that's the point where you want to be as lean and mean in terms of your proposal. But I do have to say that when you have a drug like prosetin it's a small molecule compound, it has the possibility of hitting what we call off targets. It is a kinase inhibitor, and at some concentration or another, it's going to have toxicity. So the purpose of those tox studies is to try to make sure that, for small molecule agents that are metabolized and they can convert through metabolism, either in animals or humans, into more toxic or less selective chemicals as they get mutated in the body by metabolism. Small molecule drugs are an area that less accelerations are available. because we do have to use what we call a more empirical approach. We have to study 2 species before those the FDA will even consider moving forward with a small molecule drug. So we have to look at the union set of information coming in from both a rat and a large model, and we have to say that we understand it really well and what the toxic concentrations are and what they aren't in 2 types of mammals. And then we carry that forward and say that we understand well enough to move forward. Otherwise, we're fairly convinced that if we didn't do that toxicology work, it would possibly hurt people. And I think you also have to understand that the very first studies that are done in humans are usually done in normal volunteers. That is in order to bridge the gap between animals and humans, and it's to understand the pharmacokinetics of the drug and how quickly it can be cleared in the human system. That guides the dosing regimen for humans. So it has to go stage-wise, first from animals, then into usually normal volunteers. We get the pharmacokinetics in humans. How long does the drug last? How often do we have to dose? Was it tolerated at the doses we thought were appropriate from animals? Check that box. Then we move into trials in ALS patients.
So we are skipping that healthy volunteer step for prosetin, because I think that's one area that our team feels, including our clinical team, that we can sort of cut the fat off the program and move things forward a little bit.
Well, that's where you're going to need all of the folks that are on this call and all of their friends to step up for that study, because it's a very important research space. You have to understand that when you engage the patients in that first step, the very first doses are not going to be the ones that are active. They're going to be pharmacokinetic dosing strategies. You've got to learn if it's tolerated. And then the idea is, can you offer those patients that volunteered their time and energy, and let's face it, blood and urine. That's what those studies are about, people.
Life-born.
Please go ahead and volunteer your time to that study, because it provides that key data for how often do we dose and what kind of levels can we develop that are safe. And there's lots of protections built into those studies. So please do enroll for that, because we hope that you could re-enroll in the later trials when we get all of the active doses worked up.
Yep, definitely. So we've just gotten very specific talking about blood and urine, so maybe we can take a little back. So I think sort of this is like the existential question for you, Lauren, because as Valerie and I said at the top, and as you've sort of described, you've done many different things in your life. You've been a bench scientist, you've been at the FDA reviewing applications from drug companies, you're at Charles River guiding drug development now. But you've always been focused on these incredibly high unmet medical need indications. So places that are devastating diseases where there are really no meaningful interventions available. And I mean, I think maybe you could speak a little bit to why that is and what drives you there, but also are there sort of guiding principles, given that this is your focus in your career, that sort of help you make decisions about how to move drugs forward, or other important considerations when you're in these very serious diseases?
Yeah, I have had an unusual career that way. When I first started off, I spent my 20s in a laboratory, people. You know, I didn't read the newspapers. All of a sudden, the AIDS crisis was on us. I wasn't reading the newspaper. I was going to the lab 18 hours a day. I stepped into FDA into the middle of a public health crisis. I'm just like, oh my gosh, I'd heard about this, but I didn't really how bad it was. You know, you really have to raise your head and smell the breeze, you know, to find out what is going on in the world. And I really had been stuck in the lab and wasn't paying attention. I was young. Yeah, right. And when I realized that AIDS patients had no drugs that were active, it's not too different than today with ALS, right? Mm-hmm. It was a shock to my system, I can tell you. And you had to know somebody. It wasn't everybody on the street that had AIDS, you know, but it was prevalent in certain communities, and it was affecting our blood supply, and it was growing, and it was not detected well. A lot of things resound with our COVID thing today, right? We didn't understand the immune system enough, like COVID. You know, it's just like a lot of these things that we're hearing. We were without key data. And it really emphasized to me the importance of basic research. basic immunology research, basic neuroscience. And the kinds of things that I've been working on at NIH were directly applicable to the drugs that were coming down the pike. Toxicology is sort of its own discipline. You know, animal science and veterinary science is sort of its own discipline. But the key, like poisoning of neurons in a dish and seeing poisoning of neurons in people, it doesn't take a big leap of faith. You know, you can see what's going on under the microscope and you can see them, you know, responding to changes. It doesn't take a lot of imagination to fill in the gaps. So I worked on transplant drugs. Later, I was a mom. I had a child who had a very, very, very severe congenital malformation. And so all of a sudden I saw the side of being in a very risky, high-risk hospital and having a patient not respond to therapy. We went through 2 major operations, one of the best hospitals in our country, and lost. We lost that second child, and I went on and had a third. I've been through a lot of stuff with that. So I feel for everyone that's lost their friends and family. And I think I have wrapped that experience, which was now 20 years ago, full circle back around into kind of some of the consulting I'm doing. Because when people don't have a solution, it is an overwhelming need. It's an important one. It's something that we should focus on. And we should not let all of these needs lose focus, despite the fact that we have these other problems like COVID right now. We need to keep working on all of them, whether it's in the university labs that are struggling right now with COVID closures, or at Charles River Labs where we're full on open. We have been moving some of these programs from places like universities into our labs so that the research can continue. We've got people working multiple, round the clock shifts trying to help research.
Yeah.
Well, it's in large part due to you, Lauren, and Charles River that, our prosetin program, while slowed slightly, is pretty much on track.
Well, and I think, you described to me that you've had some learnings and that some of the experiments have had to have some additional research around them. At the very beginning stages of drug development, there's a certain amount of, going back to the blackboard and reinvention that has to occur. That's part of the learnings. A drug is, when we do drug development, it is new science, right? We're putting a new chemical into the body. We learn new things about the body and the process. Mm-hmm. And so whenever it's sort of like writing a new book chapter every time we do a drug. So I think we have to take those learnings forward. Once we get through the very first stages of doing drug development, it goes faster.
Yeah, it's really cool. I think people who are watching this should understand, you've outlined a number of, sort of hoops that a new drug needs to get through in order for it to arrive at a patient trial. And prosetin, people should know that the steps that Lauren has outlined here, many of these hoops prosetin has proceeded through already with people like Lauren guiding the way and Team Prosetin guiding the way. It's really intricate and it's really complicated, and Lauren Black makes it feel simple and doable, and she continues to inspire. Sorry, I kvel, I go a little overboard. I'm the emotional one here, but I tell you,
Well, it helps to have 17,000 people that are very dedicated. I think that one of the things I wanted to highlight is that people that work in drug development and laboratories, it can be everyone from the folks that are involved in what we consider the backroom work. They're behind the scenes, but there's hundreds and hundreds of them working on drug programs. People have to label tubes and inventory the freezers. Folks have to purchase the supplies and run the air conditioning for the animals. They've got to take care of the feed supplies and monitor the equipment and keep it calibrated. All of those people appreciate knowing that they're helping drug development move forward as part of their daily, work-a-day work. They go home at the end of the day, they don't necessarily know what drug they're working on. But
Yeah,
when
but
we hear
we get that
folks-
feeling from you in Charles River. There's such a team spirit that we built on our way through this prosetin journey.
I think,
I think that's unusual. It's not typical. It's not typical.
You guys came and talked to us. That's one of the things that we like it when it's two-way. When we try to talk to drug developers, whether they're big or small or new or old to drug development, we try to encourage them to come in and give a presentation. You guys gave that to our study supervisor team at Mattawan, and it made a big difference in how people felt about your project. And they responded to you talking about the patient's needs. And so I think that's something that we need to do more often. And we always encourage even our big pharma clients to come in and tell us exactly what they're doing in the weeds, so that we understand that people go home and tell their families, hey, I helped with ALS today.
Yep.
I talked to Sam earlier, and we've got a bunch of families that are 3 generations in this company. And so what it is, granddad started talking about how they felt their job was valuable. And then the kid got going, and then that kid had kids, and then now they're working for us.
And so, and Lauren, by the same token, the Project ALS family feels like it's grown exponentially just getting to know you in Charles River. It feels like one movement to us toward prosetin and other potential drugs for ALS, and we're indebted to you guys, and really look forward to pushing this over the finish line.
Well, it'll keep moving at I think a faster and faster pace. Once you get past those very first experiments that you've been doing, then you arrive at those, like I said, that magic exposure number that has the right non-toxic effect. And we always want to remember the highest dose that is non-toxic, because that becomes our top dose in our studies for humans. Yep. So I think that once we get past those experiments, which are a little grueling at first, then we stop the iterations and we can move forward in what we consider the IND supporting work. And that is much more cut and dry at that point. So I think your timelines will be right on your expected Gantt chart. Project managed, making Erin happy.
It's hard to do, Lauren. It's hard to do.
My colleague, Dr. Sam Schwang, is really riding herd over your timelines. And I just wanted to acknowledge his efforts, because he's knee deep in the weeds.
Thank you, Sam. Thanks to all of you. Erin, what else is going on with Dr. Black that we need to know about?
I think we've really gone through a lot. There are a couple of questions about timing for the specific program. I guess this is one question that I'll just throw at you, Lauren, although I think it's going to be a little bit more theoretical. So when you talked about fast and slow progressing ALS, there's a question from Paula on Facebook about whether fast or slow progressing ALS could require different dosages of medicine, like whether you could sort of define a disease around dosing, or whether that's sort of a different consideration around toxicology. I'm not sure, but I'll just ask you, since you're the expert in toxicology, not me.
Well, I think you can, if we look at it, like, let's take an analogy to chemotherapy, okay? If we look at the kind of drugs that we know from antiquity have often been used for cancers, we know that they're very toxic, right? And make our hair fall out or whatever, that we're treading on ice, thin ice with those. They, particularly folks that know about chemotherapy drugs, know that a lot of times they'll cause us to be anemic or lose our white blood cells or our neutrophils that can fight infection. They can make us immunosuppressed. But we have to take on those high levels of the drug in order to kill the tumor.
Right.
I'm not an ALS expert, so I'm going to use the cancer analogy, right?
Yeah, it works.
Okay. Well, a lot of people know somebody that's been through this, right? Mm-hmm. So I think that you will take on a drug that has a higher toxic potential, and you'll also be able to use higher doses of it that are close to the tox threshold.
Right.
Because we push cancer drugs all the way up into toxicity, right? And then a lot of times, if your blood cell counts drop too much, they have to back the dose down a little bit so that your blood cells come back up. That's called titration. And that kind of principle is definitely applicable to some of the more aggressive cancers. So there is a connection between dose and the achievable the risk-benefit balance for a fast progressing or very acute onset ALS situation. You would be willing to undertake what you might call a higher dose or more toxic level of a drug in order to defeat that. You would be willing to take on more risk in order to balance the aggressiveness of the disease.
Yeah, that makes sense. I guess
each drug is going to have its own dose thing, though. You know, that has to be individually characterized.
Well, of course, of course.
Mm-hmm.
I want to remind everybody, I'm going to remind everybody that today is Giving Tuesday. At Project ALS, the board of directors has really stepped up and offered matching up to $10,000 today only. So if you're so moved, and many of you already have been so generous, please help us to get prosetin to human trial in 2020 by going to prosetin2020.org. And Lauren, I also wanted to invite you to another prosetin fundraiser that we're going to be doing online this week. This is going to be trippy, you guys. So Project ALS has a little spinoff called Women and the Brain, because we feel across neuroscience, you know, women really need to start to pay attention, not across neuroscience, across women's health. You know, we've gotten pretty hip to reproductive health and how to take care of our hearts even in recent years, which has been really helpful. How to do our, you know, breast exams when we need to do those. But what about the brain? You alluded to the decade of the brain having been somewhat lost. I mean, it kind of made its way through the shadows. But we're trying to pull it back into the spotlight, and we want women to talk about what makes our brains tick. Everyone wants to know. And so on Thursday, this Thursday.
Except the guys, never mind.
Except the guys. Well, you know, if you're a guy and you want to tune in, tune in. But,
please,
you know, if you're
please,
curious,
we want you to, know
that.
These things.
But for all those who want to participate in this year's Women in the Brain, it's a virtual event. It's happening this Thursday at 8 p.m. Eastern Time. It's starring Judy Gold and Rosie O'Donnell, 2 Emmy Award winners who are going to give us some comedy. It's starring sommelier Sally Stewart, who has created a cocktail just for this event. And, you know, I guess you'll see a couple of us shift in and out of the picture, hopefully to the minimum. And, but so sign up. I mean, Lauren, I'd like to treat you to a ticket. But for
if I get the cocktail recipe, I'll be happy.
Yeah.
Wait, say?
I want the cocktail recipe. Will it be made
You're going to
public?
get it. So here's the cool thing. When you register for this, you receive not only a confirmation of the registration, you receive the recipe for the Sally Stewart cocktail, which is called Pomerita. You got to be careful. There's antioxidants in it, and there's also some other stuff, not just antioxidants. So anyway, so go to the chat box, you all, to get information for how you can register for Women in the Brain this Thursday. All proceeds to support prosetin getting to ALS trial in 2020. I want to thank my colleague, Erin Fleming, who has her eyes on someone's question, perhaps? No?
I had my eyes on the chat box.
Ah, the chat box. Well, I hope it's working.
Yep, the link is up now.
I wouldn't know. I'm a techno-boob. But anyway, I really want to thank Erin and Dr. Lauren Black and Charles River for, I don't know, just helping us to feel so encouraged during tough days that there is help on the way. And you're an abiding force in our efforts. And thanks for joining us today. We look forward to reporting back to you guys next week with our live stream. Surprises there. But we'll next see you this Thursday night at Women in the Brain. Lauren, thanks so much for joining us today.
Bye, everyone.
A pleasure.
Bye-bye.
Thanks for having me. Bye-bye.